Good afternoon, everyone. It's lovely to be in Bologna, and this is the first time into this part of the country. So first of all, I'd like to express my gratitude to the organizers of allowing me to represent my supervisor, Dr. McFarland, to deliver this talk today. So what I'm going to talk about today is really the epidemiology studies that we have recently published, and also to discuss about the development and utilizations of our UK mitochondrial disease patients cohort. So in case that you haven't been to Newcastle or northeast of England, so this is the Tyne Bridge or Tyne site, and so they're connecting between the two cities. This part is Newcastle, and this is Gateshead, and this is the Tyne Bridge. And this is the Angel Knoll, this is Bamborough Castle, and some of you might recognize this castle because this is the Enric Castle that was filmed in the Harry Potter films. So I'm going to structure my talks into three components, and so I'm going to talk about the epidemiology of adult mitochondrial disease based on the Northeast England's experience. As we all know that mitochondrial disease can affect any age group. It can be caused by genetic mutations either in mitochondrial DNA or nuclear genes, but also it can affect any systems because in theory you can find mitochondria in all cells except the red blood cells. So it is very diverse topics, and as the speaker elucidated earlier, that definition of mitochondrial disease hasn't really been revisited since the initial discovery. So traditionally, northeast of England has been studying mitochondrial disease for a long time. And so when I say northeast of England, I refer to mainly 4 counties in the northern part of the UK. So there are 4 main counties, which is Northumberland, Tyne and Wear, which is where Newcastle is located, Durham, as well as Tees Valley. So according to the national census data in 2011, the population size in these 4 counties is more than 2.1 million. So the first epidemiological study was published in 2000, and the second one was in 2008. And you may wonder what is new about this 2015 one. And really there are 2 new features in these current prevalence studies, and because this time we included the prevalence of nuclear genes, which was not previously included. So the methods really is, so Newcastle is the single specialist mitochondrial center located in the northeast of England, and receive referrals from various sources from pediatricians, ophthalmologists, cardiologists, and other medical specialties. And in these studies, we only look at adult patients who are aged 16 years old or above. Compared to the previous study that only looked at the working age, this time we don't have an upper age limit. And obviously, in order to narrow down the definitions of mitochondrial disease, that need to be known pathogenic mitochondrial DNA mutations and known pathogenic variants in the nuclear genes, or clinically and biochemically there's evidence of mitochondrial disease, and also molecularly there's evidence of multiple deletions, but genetically hasn't been determined. And of course, for the epidemiology studies, we need to have very comprehensive family tracing as well as testing whenever possible. And the denominator for this study is 2.1 million of the individuals who live in the northeast. So how do we define clinically manifesting mitochondrial disease rather than just mutations carrier, but actually clinically unaffected? So these are our inclusion criteria for our study, as you will be familiar, such as the progressive axonal hemiplegia, ataxia, or other organ involvement such as cardiomyopathies, diabetes in a known causing mitochondrial DNA genes. So we do not include or we do not define any individuals who only have isolated myalgia or migraine, even if they harbor the mutation, because these symptoms are so common in the population by its own. So the result is for the mtDNA mutations, the prevalence of the clinical manifesting mtDNA disease is 1 in 10,000, which is consistent with the last study. So we also look at the at-risk individuals, so it's about, again, 1 in 10,000. So the prevalence of the mtDNA disease in the northeast of England hasn't really changed over the last 10 years, so it remains 1 in 5,000. But what is new for this study again is we have looked at the nucleogens-related disease. So it is around 10 in 100,000 in the adult populations. And it may be slightly surprising to the audience, SPG7, as suggested by one of the speakers earlier, today actually has become, has actually possibly was under-recognized in the past about its role or its link to mitochondrial disease. And other mutations in nucleogens that we look at, including the autosomal dominant PEO1, OPA1, polymerase gamma, and etc. So all in all, combining the mtDNA nucleogens related disease, the prevalence is 1 in 4,300. And I believe that this number is going to be revised constantly, and because until we actually define what exactly is mitochondrial disease, any sort of secondary change in mitochondria may be lumped into the figures. So that is the epidemiology. I'm just going to talk about the development of the UK mitochondrial disease patients cohort. Again, the background picture here is the time bridge. So I think that I do not have to convince the audience here why is it so important to develop a mitochondrial disease cohort, as clearly, whilst we know more about the molecular and biological mechanisms of the mitochondrial disease, there is still really lacking of natural histories of patients, how their disease progress, and this is what patients really ask us for whenever they attend the clinics. And because of that, in the past, there are a few randomized trials or open label trials have been conducted, but because the natural history of disease is not clearly understood, so patients may be selected because they are actually at a different stage of the disease, and therefore the drugs may appear not working simply because the patient selection is the problem. And as a result, currently, I think it would be fair to say that worldwide there is no evidence-based guidelines for patients with mitochondrial disease. So in 2009, the 3 nationally commissioned centres in the UK, namely Newcastle, London and Oxford, and so they formed the steering group committees and say that we need to address these challenges. So Newcastle led the mitochondrial disease patients cohort project, and this person here, Dr. McFarlane, is the chief investigator. So the patients cohort project was started in 2009, and last year we have renewed the ethical approval and allowed this to become, instead of just a patient registry, it is now a longitudinal prospective studies. So the inclusion criteria for this cohort project is very simple. So they need to have a clear diagnosis of mitochondrial disease, either genetically or biochemically. So again, respiratory change dysfunction is important and diagnostic criteria. And there's no age limit for including the patients. And it's also important to stress that this is a living cohort. So for example, if we receive a sample and the child died afterwards, then we wouldn't count it as the patient as part of the cohort. But if the patient died during a follow-up, we'll retain the data. And perhaps one thing to mention what is unique about our cohort project, or at least in Newcastle, is we incorporated the mitochondrial disease rating scale as part of our routine clinical assessment. And so this rating scale was published in year 2006, and really it has served 2 main purposes at the beginning. One is to help the clinicians to streamline, to define the multi-system involvement of mitochondrial disease clinically. but also actually if you then subsequently perform it on the subsequent follow-ups, you can use that as a disease progression assessment tool. I think that that's emergence that clinical trials is forthcoming to mitochondrial disease patients. So this can potentially use as outcome markers. So just a very brief word about MDOS. There are 3 sessions, 29 questions, and it covers various systems, but neurological systems is heavily weighted as you may appreciate. Neurological involvement remains by far the most common system affected by mitochondrial disease. This is just examples of the MDOS questions of trying to grade the severity of ptosis. You may argue that, there's no numeric figures here to say how many millimeters the pupil is obscure, but we also have to factor in that this is also a clinical assessment tool, so you have to trade in between the, is clinically easy to use versus very thorough but very time consuming. So this is just pictures to show that ptosis, yes, it's the clinical features, but ptosis comes with various severity, and therefore it's very important to grade the severity of ptosis, especially if you're going to do a trial. So so in our mitochondrial disease patients cohort, the database have 2 components, and one component is the patient database, which simply we keep or record the patient demographic data, their names, their date of birth, and their address. And in the orange database, or a clinical database, is where we store wide arrays of clinical information, ranging from just height, weight, blood pressures, to genetic diagnosis, clinical phenotypes, the MDOS scores, and imaging, and etc. So just to give you an overview of what have we found so far since the project started in 2009. So this heat map here do not represent the epidemiologies of the mitochondrial disease, rather it just reflects on where the patients were recruited. And as you can see, northeast where Newcastle is, we have a very red hotspots there, and the population there is only 2.1 million. So if we really go based on population density, we should really see the big hotspot around London, or in second largest city in the UK, Birmingham. But in these 2 areas, that we believe that mitochondrial disease remains under-recognized. So if our estimations of prevalence 1 in 4,300 is correct, we would really expect there will be around 6,000 to 6,500 mitochondrial disease patients in the UK. So just to break down in terms of the individual centers recruitment, so in Newcastle we recruited nearly 700 patients in the last 5 years. So the next few slides, I'm just going to focus on data collected in Newcastle, and this graph here basically just shows you the age distributions of our patients. As you can appreciate, it really covers from very young children to very elderly adults. And so 6.6% of the patients died during the follow-up, and this is predominantly an adult cohort. As you can see, nearly 90% of the patients are adults or those aged above 16. In terms of the breakdowns of the genotypes, while we appreciate that there are currently more than 200 nucleogenes being linked to disease, but by far, in terms of clinical caseloads, the 3243A2G mutation and mtDNA remain the most common pathogenic mutations. And I think this is actually similar to the Italian figures, about 30% I gathered earlier. And the second commonest cause of mutations is the sporadic single large-scale deletions, which is around 15%, which is fairly consistent with the Italian figures of 19%. And so, unsurprisingly, perhaps for the audience here, in adults, the prevalence of the mtDNA disease, so it occupies about 2/3 or 66% of all cases, whereas nucleogenes in adult patients only occupies about 1/3 or 33%. And in our cohorts, currently genetically undiagnosed cases are around 10%. And this picture here is basically just the breakdowns of the clinical features in adult patients. And this is, so this graph is prepared based on the MDAS scores. So the dark blue bars here basically represent neurological involvement, either it's central or it's peripheral. And as you can see, in adult patients, ptosis and CPO are perhaps the most common presenting features, and myopathies and cerebellar involvement. But it's important to also highlight how common or how prevalent other system involvement is. So deafness presence in about 45% of patients, GI symptoms, as suggested by other speakers, is extremely common, and diabetes mellitus and cardiovascular system, because in our experience in adult patients, patients do not necessarily die as a result of neurological involvement. However, cardiovascular involvement is the major killer. This picture here is the dot plot, and basically just to give you at a glance what is the disease burden across different genotypes. And so each dot represents one patient. As you can see, even between the same genotypes, the disease severity varies a lot. But when you compare between different genotypes, then you can see that actually those autosomal dominant nucleogen-related diseases actually are relatively mild. So based on that, actually, that gives us quite firm information when we discuss with the patients. We can say that, look, not everyone with 3 to 14 mutation is going to have stroke-like episodes, and small proportions of them will have it. But I guess the important question now is, are we able to predict who will develop the severe phenotypes? So just probably carry on talking about 3to4-3A2G mutations, since it's the most common and important mutations. And this is basically the age range of patients. And I think possibly quite similar to what the previous speaker says, we also have more female patients than male patients. I guess for mPDNA disease, female patients normally would have more concerns about its implications passing on to the next generation, therefore more willing to seek help compared to the male patients. In terms of the phenotypes and distributions, this is based upon about 220 patients. And the most common involvement in terms of system-wise is gastrointestinal. The traditional acronyms of MELAS or stroke-like episode only affected 20% in our cohorts. And so diabetes is commoner, so we would say we will have more MEAT phenotype patients compared to the MELAS phenotype. And again, it's very important to stress that when we do counseling to patients and family members. And so this is just to show that other than the clinical features, we also retrospectively and prospectively collect brain imaging, and it's quite nice that when we are able to put all the scans next to each other and see how many stroke-like episodes an individual has, which part of the brain the patient has, and what is their disease burden, and what is their current MDAS score, for example. And this is just to highlight that for GI involvement, yes, some indigestion, constipation is very common, but during the data mining, when we look at severe gut motility or pseudo-obstructions, we actually notice that it affects. So among those with MELAS syndrome, so it affects half of them, and most of the time this pseudo-obstruction develop peri-stroke-like episodes, and possibly 1 week or 2 weeks down the line. But actually, because stroke-like episode is so dramatic, maybe we've just been so drawn to look at the brain, but actually neglecting until these pseudo-obstructions creep in when their neurological system improve. This is just an example of how we can chart the disease progressions using the MDAS and looking at different heteroplasmy level. And so I have talked about the briefly discussed about the contents of the disease cohort. Now I'm just going to give you some examples of how we have utilized it in clinical practice. So since 2009, so this is a cohort project that we always foster and encourage collaborations either nationally or internationally, and that is the committees that will oversee the applications. So to date, 28 studies have been approved, and they can be broadly divided to either a data mining project or actually use the cohort as a registry to screen for patients, because our patients very well phenotyped, so many researchers benefited from that. And perhaps just briefly touch upon about cardiac involvement. As one of the speakers mentioned earlier, cardiac involvement is a major cause of morbidities as well as mortalities. So in the adult patients, nearly 20% of them have cardiac involvement, but I know that there's always argument that is always loose genotype-phenotype correlations, but believe actually that is still a very strong correlation, at least in the cardiac phenotype perspective. And because, as you can see, that the cardiac involvement is very rare in adult nuclear gene-related disease, but when you look at mtDNA disease, when you look at single deletions, they are very prevalent. And this graph here just show you the breakdowns of the types of cardiac involvement. So for 3-4 mutations, for example, hypertrophic cardiomyopathy is way more common compared to, let's say, in single deletions, which the cardiac phenotype is really the conduction block. And yes, some patients would have dysfunctions of LV functions in single deletions, but we believe that's more because of conduction defects rather than because of a primary cardiac muscle problem. So these are just the examples of the utilizations and led to increase our knowledge about mitochondrial disease. And this is the papers talking about the disease progression in single deletions. And this is the questionnaire survey study. This is just to look at the urine proteosomes of patients with mitochondrial disease compared to normal populations. And there's a cardiac study, that is gait analysis, and there's also a neuropathological study for disease patients. And so the disease progressions modeling is currently freely available in the web page, and we encourage if any clinicians who have patients single deletions, please feel free to visit the web page. And essentially the data you need for analyzing or to use it for modeling is really you need to know the breakpoints of the deletions and the deletion size. And recently, together with Dr. Gorman as well as John Grady, we have also developed the cardiomyopathy modeling just to look at what age the patients at risk developing cardiomyopathy, and then that would actually inform how you should or how one should consider in terms of the surveillance strategies. And so I suppose what we all share here today is we really want to develop the best possible clinical care guidelines for our patients. And currently we have developed 8 guidelines and it's 2 are in progress. So I think the future directions now is that every part of the world have patient registries and the numbers are increasing and patients are getting better and better phenotypes, and actually we're probably moving towards the eras of clinical trial readiness. And so if there's ever a substance that proved to be potentially benefit with the availabilities of the registries, I think patient recruitment shouldn't be as a bigger problem as in the past. And just the last word about the Cochrane review and discussing about treatments in mitochondrial disease. When I first joined the research group, I find it rather depressing because despite there's so much knowledge about mitochondrial disease, there is no single treatment seems to be clinically meaningful to the patient. there might be some biochemical evidence of improvement, but in those studies look at the trials, none of them seems to respond and report clinical responsiveness. And I suppose that is just a reminder for us that we really have to move on. So this is my final slide, and I just want to acknowledge all the members here. And this is Professor Turnbull, and this is Professor Taylor, this is Dr. McFarland, and some of the people here are clinicians or physiotherapists or diagnostic lab scientists. So thank you very much.
Thank you, Dr. Hu. The presentation is open to the discussion. Please. Nani?
Thank you very much. It's very impressive, but I think that your data are very similar to our data. So meaning that we both have probably same methodology. I would like to have more info, if you have a chance to give me that, about the heteroplasmy and how do you really use in predictive model the heteroplasmy in the natural history of disease, because when we were looking at our cohort, we didn't really find any correlations, while in your data, frequently you do have correlation between heteroplasmy and natural history. So this is disagreement between our two studies. So what's your impression?
And so that is a very good and challenging question to answer. And I think for the single deletions, the combinations of heteroplasmy level and mutation and the deletion size have clearly shown the associations between disease burdens as well as the two factors we discussed. For the 3243 A to G mutations, and I think this is quite challenging because we have seen lots of mixed data from different groups, and some groups seem to suggest that perhaps there is a heteroplasmy level correlation with disease severity, or perhaps with certain type of phenotypes. And what I can say is we do see that the heteroplasmy level, for example, referring to blood and in 3243, for example, seems to have good correlations with cardiomyopathy, and also males seems to be affected by cardiomyopathies at an earlier age compared to females. So again, that possibly will go back to in a liberal story and experience, as you also suggested in your recent paper about whether that gender does play a factor in Miller syndrome. So I think that what we are saying is we don't think this is conclusive, but we just try to find better way to develop better models to more robustly predict the progression.
Yeah, well, I was wondering if we should actually input our data in your predictive tools and see what happens, because it seems that you have some algorithms that are able to project what's going to happen to the patient based on the data that you input into the system. Is that right?
Yes.
And I'm not clear about, are you putting into the database also preclinical patients, people from the family which are carrying the mutation? Essentially they're not affected, or they're affected only if you go and look instrumentally without examination.
I think you're right. So for the inclusion criteria, so we do have patients who are unequivocally clinical symptomatic patients. I think the definitions of asymptomatics really is up to debate and discussions. For example, we know most of the cardiomyopathy is subclinical, so they could be asymptomatic, but actually they should be classified as clinically affected. So we do. So bearing that in mind, so we do enroll patients with mutations, but they haven't reported symptoms. because we also believe that actually that will serve as a very good platforms to follow them up longitudinally. Then you know at one point they start to develop symptoms, so that will give you a prospective data rather than retrospectively we go back to questionnaire survey and say, do you remember at what year you developed these symptoms?
So it will come a day that we do a meta-analysis of both.
Yes.
Series.
Yeah.
Okay, no more questions? Thank you.
Thank you.